to introduce Larry Moran. Uh, Larry is a biochemist and evolutionary biologist, and he's also one of us, senior college fellow. So Larry uh, pursued undergraduate education at Carleton before moving to do a PhD at Princeton. He then undertook 4 years of postdoctoral work at the University of Geneva and then joined the faculty of the University of Toronto in the Department of Biochemistry. His research has focused on the regulation of gene expression, heat shock genes, and molecular evolution. He's the co-author of the Principles of Biochemistry, a textbook that has seen multiple editions. He's also the author of a science blog called Sandwalk that he's been publishing for many years, and I noticed that he's brought us a slide of this. So I thought that was a strange name for a blog and checked it out. And I was delighted to find out that the name Sandwalk refers to a pathway. I'm sure that that's the one we're seeing here. It's a pathway behind Charles Darwin's house, which Darwin himself created and planted and on which he walked almost daily as he thought. So it turns out that Larry is a great admirer of Darwin, and he's called him the greatest scientist who ever lived. So to those of you who might say, well, what about Isaac Newton? Larry offers, amongst other things, a rather provocative response saying, well, biology is much harder than physics. And I think a couple of people in the audience may take umbrage at that. In 2023, Larry published a book with University of Toronto Press called What's in Your Genome? 90% of your genome is junk. And this explores the controversy around junk DNA. And I think we're going to hear more about that today. So Larry.
Thank you, Michael. You stole a little bit of my talk here because I wanted to start off uh, by saying that we're going to be talking a lot about biochemistry, yay, molecular biology and genetics today. But I wanted to start off by showing you this pathway, this sandy pathway. And as he said, it was the pathway behind Down House in. Is that better? It's the pathway behind Down House, south of London. That was the home of Charles Darwin for 40 years, from 1842 until his death in 1882. And he used to walk on this path every single day. He called it his thinking path. And it's where he developed his scientific ideas and dealt with the issues of the time. It's where really natural selection was developed and expanded. And I bring this up not only because my blog is named Sandwalk, but also because we're going to be talking today a lot about evolution as well as biochemistry and molecular biology and genetics. So let's make sure we're all in the same path when it comes to talking about your genome. Your genome is all of the information that's found in one copy of each of your chromosomes. Okay, so where were we? Oh, yes. So we're making sure that everyone understands what a genome is. The genome is one copy of each of your different chromosomes. We are all diploid organisms. That we have two copies of every chromosome, but the genome is only one copy. So there's 22 autosomes, one X chromosome, one Y chromosome. So if you're going to describe the complete human genome, it's all 24 different chromosomes. The chromosomes that you see in this image are complex structures of DNA and protein, but all we're interested today is the DNA component. What's in your DNA is basically what we're going to be talking about. Your genome contains 3.1 billion base pairs or units of DNA, and that's a fact that we've known for 3/4 of a century. Now, I'm pretty sure that all of you know something about DNA because you all took biochemistry in university, right? Yes, yeah. But just in case you've forgotten, here's a bit of a refresher course on what the structure of DNA looks like. It's DNA is just a long, skinny molecule. It has two strands that wrap around each other to form this famous double helix. And the business part of the molecule is the part in between that's formed from base pairs. There are only four bases. Let's just call them A, T, G, C. That's all you need to know. And there are pairs of them inside the sequence. Now, Watson and Crick's model of DNA back in 1953 turned out to be very remarkably accurate. They calculated that the space between these base pairs would be about 0.33 nanometers. So that means that the length of your complete genome, if you put all the DNA molecules end to end, would be about 1 meter. But since you have two of them per cell, so that's about 2 meters of DNA in every cell of your body. So that's what I mean by a very long, skinny molecule. You can't see it, of course, even though it's that long. So all of this DNA is packaged in the nucleus of your cell. The nucleus is a spherical membrane body within your cells, and all the DNA is inside that membrane sphere. Bacteria don't have a nucleus, but we belong to a very much larger group of organisms that evolved later on. We have more DNA than bacteria, and it's confined to the nucleus. So we are eukaryotes. Bacteria are prokaryotes. These are the few scientific words that you're going to have to learn in order to follow along. So you've got about 10 trillion cells in your body. So that's about 20 trillion meters of DNA, or 20 billion kilometers. That's about 4 times the distance between the sun and Pluto. So all the DNA in your body. So what's it all doing? Okay, that's the question for today. When we talk about sequence, we're actually talking about the only important part of the DNA molecule is the sequence of these various base pairs. That's where all the information is contained. So when we're talking about sequence, we're referring to the order of these base pairs in DNA. For example, the sequence on the slide is read from on the left-hand side from top to bottom. So you can define this DNA molecule by simply saying it's ATGC. Okay, then everybody who's taken biochemistry knows that that's what you're talking about, and that's the important part of the information. It is that sequence. That's why we talk about the sequence of DNA, or the sequence of base pairs in a molecule. The sequence is stored. We now know the sequence of the human genome, and it's stored in a database that looks like this. Note that you only have to specify one of the nucleotides or one of the base pairs in order to define the molecule. So the question is, what do all of those A's, T's, G's, and C's, 3.1 billion of them, how do they make a human being? Are they all necessary to make a human being? That's the point of that's the subject of this talk. Now, there's lots of different ways that I could give this talk in order to bring you up to speed on molecular biology, genomics, genetics. But I've decided that the best way to do it is to do a historical overview of how the field developed over the last 3/4 of a century. So I think it's best to follow this historical development because it highlights the current controversy that I'm going to get to at the end of the talk. So I'll begin by explaining why the leading scientists of the last century came up with the idea that your genome is sloppy and most of it is junk. This resulted in a model that only 10% of our genome is actually functional and 90% of it is junk. So this was the model that was proposed in the early 1970s. Further evidence in support of this model accumulated in the 1980s and the 1990s, and the publication of the complete human genome sequence in 2001 confirmed the model. It proved that the experts were right and the amateurs who were predicting more function were wrong. But the experts were ignored. Most scientists, when they saw the human genome sequence, were very uncomfortable with the idea of a sloppy genome full of junk. So they came up with a number of excuses to dismiss the evidence of junk DNA. And this culminated in a series of papers published in 2012 that rejected the model of junk DNA. The experts fought back and refuted the claims of the anti-junk crowd, but that didn't make much of an impression on the scientific community and the general public. So what we have today is a popular press that promotes a completely false view of what's in your genome, a view that conflicts with that of the most knowledgeable experts in the field. Now, this is all covered in my book, which I encourage you to read. Try and give you a brief summary of all the science behind this controversy, and then we can discuss the reasons why misinformation is so common in the popular press. Our story begins in the 1940s with a debate about mutations and their frequency. There are lots of genetic diseases that are caused by mutations. You're familiar with many of them. We can tolerate about one new mutation per generation because we have two copies of every gene, two copies of every chromosome, and you usually only need one of them to stay alive. So, so the gene variants that arise by mutation are called alleles. This is another word that I wish more people knew about. So alleles are different copies or different versions of the same gene. So if we have a mutation for cystic fibrosis or diabetes or whatever, you would generally be carrying two alleles for the same gene, a mutated allele and the normal allele. Now, the mutated allele will normally be eliminated from the population because when you give rise to children that have two copies of the bad gene, they usually not survive, right? So mutated alleles will gradually be severe mutated alleles will be gradually purged from the population. JBS Haldane was one of the founders of population genetics, along with Ronald Fisher and Sewell Wright. And the basis of population genetics is that it is populations that evolve, and the mechanism is the accumulation of mutations and their eventual loss or fixation over time. Now, loss, you're familiar with, bad genes will be lost. Fixation is when an allele or a version of a gene eventually takes over the entire population. It becomes fixed in the population, eliminating the other copy. This gave rise to the modern definition of evolution as a change in the frequency of alleles in a population. That's the version of the definition of evolution that's taught today in high school. Okay? The evolution is a change in the frequency of those alleles in a population. Haldane estimated the rate of mutation in humans by looking at the frequency of genetic diseases. His most important paper was published in 1949. That's this particular paper here. Hermann Muller was awarded the Nobel Prize in 1946 for discovering that mutations could be induced by X-rays. He was also interested in the mutation rate in humans and what effect that could have on our survival. Muller and Haldane calculated that the rate of lethal mutations in a typical gene is about 1 in 30,000. So what does that mean? That means that we can't have more than 30,000 genes, or we would be accumulating more than one mutation every generation, and our species would go extinct. So this is the mutation load argument, that the number of sites, the number of lethal sites in our genome, the number of genes can't be more than 30,000 because we know the rate of lethal mutations, and we know that we couldn't survive if we had too many of them every generation. So by the 1960s, we had a pretty good idea of the size of a typical gene, and 30,000 genes would only take up about 2% of our genome. Whoops. What was the rest of the genome doing? Most of it appears to be junk because it's unaffected by mutation as far as we knew back in those days. Now, at the same time, in the 50s and 60s, data on the genomes of genome sizes of various species began to accumulate. And it soon became apparent that there was a huge difference in genome sizes between different species, and that didn't correlate with expectations on perceived complexity. For example, the pufferfish on the left is one of the smallest vertebrate genomes. It's about 1/8 the size of ours. Now, this may seem reasonable because I think we all would like to believe that we're much more complicated than a pufferfish. So, however, the African lungfish has a genome that's over 40 times bigger than ours, and most of us don't think that we're more complex than a lungfish. Okay. Furthermore, most of these species are fish, or both of these species are fish, and they likely have a very similar number of genes in spite of the fact that there's a 300-fold difference in the size of their genome. Now, these differences in genome sizes can even be seen in more closely related species. For example, the red viscacher rat has a genome that's more than twice as big as ours, and the bent-winged bat is a mammal just like us, but its genome is almost half the size of ours. It seemed pretty clear back in the 1960s that the amount of DNA in your genome didn't correlate with complexity. And this gave rise to the idea that genome size doesn't matter so much. It gave rise to what's called the C-value paradox, where C value refers to the amount of DNA in a single cell or the size of your genome. The range of C values is usually shown in charts like the one on the slide. And you can easily see that for each group of species, each black bar, there's a huge range of genome sizes. Note that the x-axis is a log scale axis, so it's tenfold. The first thing to look at is the difference between the bacteria at the very bottom left-hand corner of this slide and everybody else. Bacterial genomes are a lot smaller than genomes of species like us. And it turns out that bacteria have very little junk DNA. Now look at frogs. That's the fourth bar from the top. Okay. There's a 100-fold difference between the size of the smallest genome and the size of the largest genome, yet they all look like frogs. Okay. So the question is, why? They have all this. Why is there this big difference? Lots of crustaceans, like lobsters, have genomes that are bigger than ours, and so do lots of plants. In fact, there are even single-celled organisms like amoebas that have genomes that are a lot bigger than ours. Okay. The C-value paradox, that is, how do you explain these differences, is a very important part of our story, so it's worth looking at how the issue was described in the textbooks from 50 years ago. In this particular case, I'm showing you Benjamin Lewin's description from the 1983 version of his textbook, but he said pretty much the same thing 10 years earlier in 1973. He says the C-value paradox takes its name from our inability to account for the content of a genome in terms of its known function. And here's the important part. One puzzling feature is the existence of huge variations in C values between species whose apparent complexity does not vary correspondingly. So this was a big deal back in the 50s and 60s and early 70s. How do you explain this? Now, these observations gave rise to the idea that a large part of these genomes, including humans, was junk DNA with no function. Now, it's important to note that the C-value paradox is no longer a paradox. It's been resolved. We know what the answer was. And so did Lewin when he wrote this in 1983. This is the introduction to his following explanation of the C-value paradox. So we'll get to explaining what what the explanation actually is in just a minute or two. But that brings me to another important discovery in the late 1960s. I've been casually referring to genes in the past few minutes. The idea is that genes, in the genetic sense, refer to some kind of function that's being carried out by each gene. You have a gene for hemoglobin, you have a gene for muscle protein, you have a gene for insulin, for example. Um, each gene is responsible for a particular trait, and each one should be unique in your genome. Thus, your genome should be full of unique sequences that specify a human being. But by the late 1960s, scientists had developed techniques to determine how DNA sequences were organized in a genome. And they discovered that a large fraction of a typical eukaryotic genome like ours consisted of repetitive DNA, not unique DNA, but repetitive DNA. That is, bits of stretches of DNA that are repeated over and over again. About 50% of your genome consists of repetitive DNA. The simplest kind at the top of the slide is the highly repetitive DNA consisting of hundreds or thousands of simple repeats like AT, 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 AT. It's pretty boring. Okay. The next category is called short tandem repeats, or STRs. This consists of multiple copies of short repeats of anywhere from 5 to 25 base pairs, nucleotides. There are a lot fewer of these repeats than with the highly repetitive DNA, but we now know that there's 350,000 of these in your genome, and they make up about 5% of your DNA. By the way, these moderately repetitive, these short tandem repeats, STRs, um, are responsible for DNA fingerprints. At any given locus, at any given site in a chromosome, some of us have 9 or 10 copies of these repeats, while others may only have 3 or 4, and some have 18 or 19. If the numbers of these repeats range from, say, 2 to 21, then there are 20 different possibilities that distinguish one person from another. If you look at 20 different sites, then the probability that any one of us will share exactly the same number of repeats at all 20 sites is extremely low, lower than the number of people on the planet. Thus, everyone's DNA fingerprint is unique, except for identical twins. Okay. So this is consistent with the idea that the amount of this repetitive DNA isn't terribly important, since we all differ by quite a bit. It's irrelevant. And that's what the experts concluded in the late 1960s. The third category of repetitive DNA is called moderately repetitive DNA. These are longer sequences of hundreds or thousands of base pairs that are dispersed in your genomes. A little bit over here, there's one over here, there's one down here. Each one may only have a dozen or so different copies, but they're big and there's a lot of them, and they make up 40% of your genome. So 40% of your genome is these dispersed, moderately repetitive DNA sequences. So it soon became apparent that much of the differences in genome sizes could be explained by differences in the amount of repetitive DNA in these species. Okay, so it was generally, there was a lot of debate about these repetitive sequences. What could they possibly be doing? And it was generally concluded that most of them really had to be junk DNA. So the important point is that the presence of junk DNA accounted for the apparent paradoxical variation in genome sizes, the C-value paradox, and a lot of that's due just to the presence of these repetitive DNA sequences. So the mutation load argument and the observation that genome sizes could vary enormously seemed to indicate that our genome was quite sloppy and contained a lot of junk. But this conclusion seemed to be at odds with the prevailing view of evolution at the time. That view favored natural selection as the only significant mechanism of evolution. And since junk DNA was assumed to be detrimental, that should have been eliminated by natural selection. But in the 1960s also saw an increase in the sequence data of proteins as well as DNA, and it soon became apparent that there's lots of variation in protein sequences between species and even within species. The proteins that you make are not the same as the proteins that I make, and the difference doesn't seem to be correlated with fitness. So we differ not only in our DNA fingerprints, but also in the proteins, the sequences of the proteins that we make. For instance, if I was to look very closely at your hemoglobin and your hemoglobin and my hemoglobin, I'd probably find that they're all slightly different, but they all work the same way to bind oxygen. So that kind of variation was inconsistent with the prevailing view of evolution at the time that assumed that there are only two kinds of mutations: beneficial mutations, detrimental mutations. The good genes would quickly take over the population, and the bad genes would be quickly eliminated. Molecular biologists looked closely at these different variants to see if they indeed were the same, had the same function. And it turns out these differences really are due to mutations that have no effect on fitness. They are neutral changes with respect to fitness. It had been known since the 1920s that most of these neutral changes in a population vary in the population or change in the population. Their frequency in the population changes by random genetic drift. The man mostly responsible for promoting this idea is the Japanese scientist Moto Komura. The article he published in Nature in 1968 triggered a major rethink of evolution at the molecular level. And he followed this up with many other articles and books. The important point is that the major mechanism of evolution is random genetic drift and not natural selection. Okay? Random genetic drift is the idea that certain mutations could accidentally become more or less frequent in the population just by chance. Okay? It had been around since the beginning of population genetics in the 1920s, but it wasn't thought to be an important mechanism of evolution. So this idea of neutral theory led to a major shift in how we think about evolution, kind of a paradigm shift in our thinking about evolution. It was fully adopted by the leading population geneticist at the time, but it was largely ignored by the general public, by teachers, and by most scientists because they were unfamiliar with DNA, biochemistry, and molecular biology. This stimulated Jack King and Thomas Jukes to publish a paper in Science the following year. We're talking now 1969. They deliberately titled their paper Non-Darwinian Evolution in order to draw attention to the major shift in the way that we were looking at evolution. King and Jukes summarized all of the data that I've been just talking about mutation load, C-value paradox, repetitive DNA. They added data on the structure of genes, proteins. And they pointed out that our genome contains huge amounts of superfluous DNA or junk. And this could be explained by neutral evolution and random genetic drift. Thomas Jukes, by the way, is a graduate of our department, the Department of Biochemistry here at the University of Toronto. So until now, I've been casually referring to genes in the genetic sense as some part of your genome that's responsible for a certain trait. This is the Mendelian gene, the definition that's used by geneticists. Now it's time to introduce the molecular gene, the definition used by biochemists and molecular biologists. It's a much more specific definition that connects a gene to a particular stretch of DNA. So here's a cartoon version of the molecular gene based on what scientists knew in 1970. The solid gray line at the top is DNA, and the rectangles represent the genes. A gene is defined as the DNA sequence that's copied into RNA to make a functional product. The copying takes place when an enzyme binds to the beginning of the gene. That's the site marked with a P for promoter. And it then moves down the gene, copying the DNA into an RNA molecule until it reaches the end marked with a T for terminator. The gene on the left produces a functional RNA molecule that carries out certain functions all on its own. The gene on the right it produces a special RNA called messenger RNA that is subsequently translated to make a protein. So the words you need to memorize here are genes, sequence of DNA that's transcribed, transcription, copying it into RNA, translation, copying the RNA or into protein. So many genes are transcribed at almost every cell. But some of them are transcribed or expressed only in certain cells. For example, your hemoglobin genes are only expressed in blood cells, and your insulin genes are only expressed in the pancreas. The DNA that controls when a gene is expressed is called a regulatory sequence, and it's usually near the beginning of the gene. Okay? So the important points on this slide are that there are two kinds of genes, protein coding genes, non-coding genes. Regulatory sequences are important functional elements. They have a role to play in your genome, but they're not part of a gene. And the coding DNA, that's the black box on the gene on the right, is only a small part of all of the functional DNA that's shown in this particular diagram. So this brings us to 1972 and the publication of two papers by Susumu Ono. The first one is the one that brought the concept of junk DNA to the attention of a much wider audience. The term had been used in the 1960s in casual conversations, but this was the first time it appeared in a paper that was widely read. Ono summarized the data that I've just been describing and concluded that 90% of our genome is junk. He also published a much longer and more detailed paper in the Journal of Human Evolution, where he was much more explicit in describing this particular argument. Here's what he said in that second paper. Evolution from the simpler life forms to mammals appears to have been accompanied not only by successive addition of new gene loci, but also by the accumulation in the genome of a great deal of degenerate nonsense DNA-based sequence. At least 90% of the mammalian genomic DNA appears to represent nonsense-based sequences of various kinds. And he goes on to say, it looks like Muller and Kimura and others were not far off the mark, and that at least 90% of our genome is junk or garbage of various sorts. This was a powerful argument. It provided an explanation for the mutation load problem and the C-value paradox. It was also very compatible with neutral theory and evolution by random genetic drift, and explained the data on the structure of genes and repetitive DNA. Now, this is a good time to remind everybody about how to judge a good scientific explanation. It doesn't matter whether you call it a theory or a model or a hypothesis, its value depends on its explanatory power. When I was preparing this talk, I did a web search for the term explanatory power, and this is how Google AI summarized the term explanatory power. It's a pretty good definition of explanatory power. So keep in mind that the importance of a scientific explanation, theory, hypothesis, model, it depends in part on how well it explains the data and the observations. When you have competing models or hypotheses, the one with the best explanatory power will often turn out to be true. Here's my friend Ryan Gregory on the Sandwalk. Ryan is a professor of biology at Guelph University, and he's an expert on genome sizes. He's going to give us a test that we can use to judge various hypotheses concerning the composition of genomes. The test is called the onion test, and it goes like this. This is the version he published with Alex Palazzo, one of my colleagues in the Department of Biochemistry here in Toronto. This was from a paper in 2014 called The Case for Junk DNA, It was published in only 10 years ago when junk DNA was under attack. But that's not the important. Here's the important part of the onion test. If most eukaryotic DNA is functional at the organism level, be it for gene regulation, protection against mutations, maintenance of chromosome structure, or any other such role, then why does an onion require 5 times more DNA than a human? Okay. So if you think you have a better explanation, explain this. Now, junk DNA does explain that. Ryan Gregory is challenging you to come up with a better explanation than junk DNA in order to explain why an onion has more DNA than we do. So far, nobody has met that challenge. Nobody has come up with an explanation that accounts for all of the data, including why onions have 5 times more DNA. Okay, let's go back to our historical. I jumped ahead a little bit. Let's go back to our historical overview. Remember, there were powerful arguments for junk DNA back in 1972, but we still didn't know much about the junk in our genome beyond the fact that there was a lot of it, and a lot of it was repetitive DNA. But we did know that sometimes viruses insert their DNA into our genome, and we did know that there were other things called transposons that did the same thing. Now, transposons are little bits of DNA with carrying a few genes that can pop into your chromosomes. They can sit there for a while and then pop out again. And they're often called jumping genes. Okay. So towards the end of the 1970s, it became apparent that there were lots and lots of these viruses and transposons in our genome. And this led to the publication of 2 very remarkable papers in Nature in 1980. You don't need to read anything on the slide except the titles of the articles. The first one is the work of Ford Doolittle and his colleague Carmen Sapienza. Doolittle is a professor of biochemistry at Dalhousie University in Nova Scotia. Second paper is by Leslie Orgel and Francis Crick, who by this time had moved to the Salk Institute in San Diego. Both papers pointed out that our genome is full of viruses and transposons whose only purpose seems to be acting as selfish parasites. It suggests that junk DNA is mostly due to the invasion of our genome by these selfish parasites. So we now know that they were correct. About 50% of our genome consists of parasites like viruses and transposons. This is the source of the moderately repetitive DNA that I talked about earlier that was discovered in the late 1960s. So there are hundreds of these viruses and transposons, and they often have cute names like Mariner, Gypsy, Piggy Bank. Others have more mundane names that named after, for instance, something called line elements, so L1, L2, L3, etc. Many of the viruses in our genome are retroviruses. So the ones in our genome are called endogenous retroviruses, or ERVs. Now, most of these viruses and transposons were inserted into our genome millions of years ago. Our relatives, the chimpanzees, have the same elements at the same positions in their chromosomes. Over millions of years, these elements have accumulated mutations, shown as the red X's in the cartoon. So they're no longer capable of acting as jumping genes or transposons or active viruses. They are junk DNA. They're dead viruses, dead transposons. These parasites and their degeneration are a major source of junk DNA in all genomes. Thomas Jukes, remember, he's the one who wrote the article in 1969, Non-Darwinian Evolution. He wrote a letter to Francis Crick in 1979, just before Crick published this article in Nature. It's a very interesting letter because it summarizes the thinking of knowledgeable scientists back in the 1970s. I've highlighted one of the most important points that Jukes was making to Crick. He says, I'm sure that you will realize how frightfully angry a lot of people will be when you say that much of the DNA is junk. The Darwinian evolutionists will be outraged because they believe every change in DNA that is accepted in evolution is necessarily an adaptive change. This suggests that anything else is an outrage to the sacred memory of Darwin. So do you know what he's talking about here? He's referring to the conflict between their view of evolution as mostly neutral mutations and random genetic drift, that is Jukes and Molecular and Crick and Doolittle and others, uh, um, and the view of most evolutionary biologists, most other scientists, and the general public. Those people still think that evolution is mostly driven by natural selection that produces organisms that are perfectly adapted to their environment. The key word here is adaptation. Okay? There's no room for junk DNA in such a worldview. It's an adaptationist worldview. So where does this adaptationist worldview come from? It comes from evolutionary biologists like Richard Dawkins. His book, The Selfish Gene, was published in 1976. That's the same time frame that we're talking about here. It was already widely popular. So let's be really clear about the word selfish here. Crick and Doolittle and their colleagues were referring to transposons and viruses as selfish DNA because it only served their own purposes as parasites. Transposons and viruses have no direct beneficial effect on their hosts. In fact, they are often harmful. When Dawkins talks about a selfish gene, he's talking about a gene, actually an allele, that benefits the organism, such as a gene that makes cheetahs run faster, or a gene that changes the coat color of mice so that they blend in with their environment. The Dawkins version of a selfish gene is almost the exact opposite of what Crick and Doolittle were talking about. So this is the conflict that Jukes was referring to 50 years ago. Dawkins has followed up this book with a number of other books that spread the word about the power of adaptation and natural selection, so that most people today think that this is all there is to evolution. In 2017, the Royal Society took a poll to see what people thought of science books, and the winner of most inspiring book was The Selfish Gene. Now, this is a great disappointment to those of us who think that the adaptationist view is misleading. It makes our work much more difficult when we are talking to an audience that's unfamiliar with the prevailing consensus amongst population biologists. So what's happening here is a clash of worldviews between scientists and experts in evolution at the molecular level and traditional evolutionary biologists who are mostly interested in ecology and field biology and obvious morphological differences between large animals. By the 1980s and 1990s, we got more evidence of sloppy genomes and junk DNA. For example, we discovered a lot of pseudogenes. Now, pseudogenes are broken genes that are no longer functional. Many of them come from genes that after a gene duplication event. So you have a gene that accidentally gets copied and duplicated. So you now have the same gene, two copies of the same gene side by side. Since you only need one of them, the other one is free to accumulate mutations and become a pseudogene or a dead gene. In some cases, existing genes acquire mutations and become a pseudogene, because it turns out that losing this gene doesn't have much effect. This is how we lost the gene for making vitamin C, for example. It's now a degenerate pseudogene in our genome, but it's still a functioning gene in a lot of other organisms. So it turns out that getting scurvy for lack of vitamin C wasn't a serious problem for our ancestors, so the gene was lost. How many of you have O-type blood? Does anyone have O-type blood? Yeah? Okay. You have two pseudogenes. Okay. So there are three alleles for the ABO gene, blood type gene. One of them makes A-type antigen, one of them makes T-type antigen, and one of them is dead. and makes no antigen, so it's O type. So you have two pseudogenes in your body. So the discovery of pseudogenes, obviously junk DNA. It turns out that we have 20,000 of these in our genome, and it's about 5% of your genome. Another important discovery in the 1980s and 90s was the existence of more non-coding genes than we expected in the 60s and 70s. There aren't a lot of these, but they did increase the number of genes somewhat. We also characterized all the regulatory sequences that control expression of genes. Turns out that these regulatory sequences are bigger than the ones in bacteria, but they still only account for about 2% of the genome. So just before publication of the human genome sequence, the knowledgeable experts were still predicting 30,000 genes or less and 90% junk. The completion of the Human Genome Project was announced by President Clinton at the White House in June of 2000. Here he is with Craig Venter on the left, the head of the private consortium that sequenced the genome, and Francis Collins on the right, the head of the International Human Genome Project that produced the sequences that we all use today. We all knew Francis Collins because he had been to Toronto many, many times in the 80s and 90s because he was collaborating with Lapchi Choi at SickKids, who had convinced him to collaborate on identifying the gene for cystic fibrosis. The initial draft of the human genome sequence was published a few months later in February of 2001, and it confirmed all of the predictions of the knowledgeable experts dating back to the late 1960s. The knowledgeable experts were correct. There were fewer than 30,000 genes, and a lot of it was junk. So the actual human genome sequence did not support any of the arguments against junk DNA. All of those arguments were falsified when the human genome sequence was published. Most scientists and most science writers were very uncomfortable with these conclusions. There were fewer than 30,000 genes and lots of junk. For example, Elizabeth Panisi, who writes for Science Journal, expresses a general opinion in an article she wrote in 2005. She and others were expecting that humans would have a lot more genes than other species because we think we are much more complex than rice. Rice is the big sequence at the bottom of this diagram. It's obvious that she hadn't kept up with the discoveries of molecular biology and developmental genetics in the 1980s. This mysterious complexity, which actually doesn't exist, must be hidden in the excess DNA that ignorant scientists had labeled junk. I call this the deflated ego problem. The human chauvinists are disappointed that our genome isn't as complex as our brains and behaviors suggest to them. They expected to see tangible evidence that humans were at the top of the heap. So the question before us is whether this is a real scientific problem or whether it extends from an incorrect understanding of evolution and development. And I'll argue that it stems from an incorrect understanding of evolution and development. So the problem isn't that opponents of junk DNA were having trouble coming to grips with the idea of a sloppy genome and the idea that we aren't much more special than an onion. The problem is that they came up with all kinds of nonsense ideas about why we should ignore the data and they invented new functions that don't make any sense in order to rationalize their viewpoint. Now, the technology developed to sequence the human genome has been used to sequence other genomes much more rapidly and much cheaper, much more cheaply, or much cheaper. When we compare the human genome with that of other species such as chimpanzees, macaques, lemurs, and mice, It turns out that only about 10% of our genome is similar to the genomes of those other species. In other words, only 10% is conserved, meaning that it hasn't changed much in the 100 million years since we diverged from a common ancestor. This is an indication of function, since that 10% has been protected from evolution by natural selection. Any hits on that 10% will be lethal and we won't see them. The other 90% just doesn't seem to matter and is free to evolve. This is powerful evidence of the idea that 10% of our genome is functional and 90% is junk. None of the evidence for junk DNA and the idea that we had fewer than 30,000 genes made much of an impression on most scientists. So a bunch of the leading labs in molecular biology had banded together in 2005 to find out all the missing DNA function. And they secured hundreds of millions of dollars in funding, and they formed a consortium that they called the Encyclopedia of DNA Elements, or ENCODE. They developed powerful techniques for detecting RNA in a variety of human tissues and for mapping the sites at which regulatory sequences bound to DNA. The results were published in several dozen papers that appeared in September 2012. The most important of these were published in Nature. The important point here is that they claim to have discovered that 80% of our genome contains functional sequences. In other words, most of it is not junk. And these papers were promoted by a massive publicity campaign orchestrated by Nature and by all of the universities where the work took place. Here's an example of a press release from the Sanger Institute in Cambridge, England. There were similar press releases from all over the world. Newspaper headlines, New York Times, The Observer, Globe and Mail, all announced the death of junk DNA. This is an editorial in Science magazine written by Elizabeth Pennisi when the ENCODE papers were published. You may recall that she was the one who expressed surprise years earlier that the human genome sequence didn't have enough genes. So I'm focusing on her because she has a prominent platform and because there are many science writers who said the same thing. So I'll skip over what she exactly said. Basically, she's announcing that the ENCODE papers were pronounced the death knell for the idea that DNA is mostly littered with useless bases. A remarkable thing happened on the day that these press releases were released. The hype hit the internet. In the age of social media, there were instant criticisms of the ENCODE conclusions. Dozens of us posted on our blogs, on internet forums, and on the main social sites like Facebook and Twitter. We attacked the conclusions and the hype and pointed out that the ENCODE workers had completely ignored all of the genome science that supported junk DNA. We pointed out that tiny bits of RNA that were detected were rapidly degraded, present less than one copy, exactly what you would expect from a genome full of junk DNA. We pointed out that just binding proteins to a site doesn't necessarily mean that they have a function. The instant feedback was so extensive that within a few days, Nature editor was compelled to respond on the Nature website. And he admitted that maybe we were somewhat exaggerated in our claim to the death of junk DNA. But it's important to give the data a lot of publicity because the researchers had done a lot of work for the past 10 years. So maybe it's only 20% of the genome is functional. So I don't want to bore you with examples of all the criticism, so I picked out just one to show you what happened. It's an article by John Timmer, posted just a few days after the launch of the ENCODE publicity campaign. It was posted on Ars Technica a well-respected science website. This is a really hard-hitting criticism of the ENCODE results. I want to save a bit of time by just not going through all of the things that he said. And this was followed up over the next few years by a whole bunch of articles in the scientific literature that attacked the ENCODE results. These are remarkable critical comments on a series of papers that were published by leading researchers in the most prestigious universities in the world. It's not something that you see every day, and it wasn't the only criticism that was published in the first few days. One of the papers is actually by the ENCODE researchers who kind of retract their data after they came under attack by a lot of these scientific publications. So did that solve the problem? No, it didn't. We're still seeing lots of hype today. Here's an example of a press release from 2019. There are lots and lots of examples like this. I'm picking this one because it's University of Toronto. It also comes up at the top of the list of articles when you search for junk DNA. Basically, this thing at the bottom, previously dismissed as junk DNA, non-coding regions were once thought to have little to offer to a cure against cancer. That's just nonsense. Non-coding DNA was never dismissed by junk DNA proponents. So this kind of article is called a paradigm shaft. First of all, you set up this false paradigm saying scientists did this and this and this. Then you show, oh, well, we've discovered a little bit of function. Therefore, we've overthrown the paradigm and started a revolution. Okay, I don't know how to get rid of this. And here's what Google says just last week when I asked them about junk DNA. And they say it's a deprecated term for regions that at one time were thought to have no function. So this is what we're up against. This is actually a fairly accurate summary of all the recent scientific literature in the popular press, but they're all wrong. So the conclusion is we have a sloppy genome. 90% of our genome is junk. Most alleles are fixed by random genetic drift. Oh, and by the way, we're not intelligently designed. So thank you very much. I'll just try and get rid of that. I'll put up another slide that you can watch during the break so we can stimulate a discussion after the break. Thank you very much.